So welcome to the State of the Science number 6. This is a new way of talking about research in the field of type 1 diabetes. And we have some global scientists today coming to us from Belgium and across the US, as well as Australia. And we thank everyone for joining. I'm going to briefly introduce our panelists, and the panelists will set the stage for the discussion. And conversation will be free-flowing, kind of like a cross-idea exchange, a town hall, and a respectful debate. So we'll pause it a couple different times, 2 different times. We're going to have a poll with the audience, and we'll have the panel weigh in on audience voting and feedback. And I'd just like to take a minute to recognize our really fantastic sponsors: Kenix Genomics, Vertex, Provention Bio, Imcyse. We have a representative here from Imcyse, and Inspiro. And these organizations are at the very forefront of cutting-edge science. And we also have Provention Bio here too today. And these organizations are at the forefront of cutting-edge science, and without their support, we could not bring you State of the Science. So you can find more about these innovative companies on our website, and you can add our Ask the Expert series, both live and archived on our YouTube channel. So just a few pro tips to submit a question, place it in the Q&A. You can submit questions throughout the presentations. Participants can upvote their favorite questions, and at the end of the session, we'll answer questions as time's allowed. This event is being recorded live on the YouTube, on our YouTube channel, and will be housed there after for further viewing. So in the interest of saving most of the time for the discussion, I will do very brief introductions for each of our scientists. They have a lot more history and expertise than I'm going to, touch on. But you can connect with them on our website in the collab, where most of them are members, or reach out directly to learn more. So first, I'd like to introduce Jessica Kenison, PhD. She's currently at AnTolRx. And the AnTolRx platform approach involves the development of novel targeted therapeutics utilizing specific self-antigens and unique tolerogenic molecules to actively promote immune tolerance. Their patented nanotechnology is designed to induce antigen-specific immune tolerance, and their nano formulations incorporate 2 integral aspects which work together to send a strong therapeutic signal. I'd also like to introduce Antoon van Oosterhaut. He is coming to us from Imcyse in Belgium, and their approach is very unique. It can be sort of considered immune deletion of specific immune cells involved in the immune reaction rather than immune suppression. And it's distinctly different from what most others in the field are trying to accomplish, sort of comparatively to the induction of Tregs. So they have a trademarked imitopes that specifically block the immune responses that cause immune-mediated diseases. We also have Francisco Leon, MD, PhD from Provention Bio. And Provention Bio is developing prevention and interception approaches targeting the spectrum of T1D. Their experimental clinical programs include a Coxsackie-B vaccine for primary prevention of T1D, as well as teplizumab, the T-cell targeted mAb for the delay of clinical T1D in stage 2 patients. It's under review by FDA for potential approval, as well as for the preservation of beta cells in newly diagnosed T1D. We have Michael McAuliffe, MD, coming to us from Australia. He's he's part of Captixbio, and that company, and Captixbio's Imida device is a permanently implanted device designed to optimize access to the vascular supply and carry a replenishable payload of therapeutic islet cells with a novel immune protective membrane. This device will not require immune suppression. So that's kind of novel. Dr. Jeffrey Millman, PhD from WashU, joins us from St. Louis. He's a biomedical engineer with over a decade of expertise in stem cell technology, and his lab uses state-of-the-art techniques to develop islet cell protocols from HIPSCs and ultimately hoping to find a cure for T1D. Mark Atkinson is here from University of Florida. Dr. Atkinson is a giant in the field of type 1 diabetes, and his research is broad in scope but directed at identifying a prevention or a cure for type 1 diabetes. And I think we also are expecting Matthias von Herrath, so he may join us. I hope he might. I hope he will. He would come to us from basically La Jolla Institute, as well as Novonordisk. So we'll see if he can join us. So Dr. Atkinson, can you set the stage for this discussion? Where are things trending in terms of a prevention or cure of type 1 diabetes? I'm asking this because the topic of today's conversation is prevention of type 1 diabetes, remission or cure.
Right. So thank you, everybody, for joining in today. That will be the first thing I'll say. And apologies that Doug was not able to join, but he's here with us in spirit. When Monica presented this idea to us earlier this year, and we were searching for ideas, we collectively came up with a number of them. But this one was one that I have circled the date on, as on the calendar, as you would say, meaning of all the things in type 1 diabetes, and we've spoke about stem cells, we've talked about biomarkers, immune therapies, et cetera. This topic today, and the one I'm really, again, just excited to hear about, is about the ability to prevent and cure type 1 diabetes. Certainly from a patient's perspective, as well as their family members, they always ask that question, when are we gonna prevent the disease? When are we gonna cure it? It's not clearly for lack of trying. We need new ideas and bright minds like those on the call. And I really just want to close the introduction by saying, I thank you that we have people both from an industry perspective, translational perspective, basic perspective on the call today, because I think this is important to see progress go forward. I would ask the panelists to not do what I just did, but keep your comments short so that we can hear for their perspectives from everybody. But let's go and find out what we think are some of the barriers and what do we need to achieve to see a prevention and cure for type 1. Monica, back to you.
Okay, thanks. That's great. I think that's a wonderful introduction, and thank you all again. Let's see if we can have each of our industrial partners sort of weigh in on how their technology has a role in sort of this sort of endeavor of prevention of or cure or both of T1D. So I'll ask Jessica if she could sort of just set the stage.
Oh, I will certainly do my best. So at AnTolRx, the way that we're really trying to think about this is to use a very targeted antigen-specific disease-specific approach. And the way that we do this is actually through a platform technology where we've developed these nanotechnologies, specifically nanoliposomes, which co-deliver two molecules, one being a small molecule designed to induce immune tolerance. So it sends out a signal specifically in these targeted cells, antigen-presenting cells like dendritic cells, macrophages, where we can turn on these regulatory signals, induce regulatory T cells, and suppress effector T cells. But at the same time, to this same cell, we're actually delivering antigen specific to the disease. So in type 1 diabetes, really we're taking peptides or whole proteins from insulin in order to target specifically the cells that are activated improperly in type 1 diabetes. And we think that by using this kind of dual approach, we're hoping that this is going to really target it, prevent any general immunosuppression, but really strengthen the effects that we're seeing. And we've seen some really strong evidence of that in mouse models. We're looking obviously to hope to bring it to humans as soon as possible, because that's really where it's needed. But that's the approach AnTolRx is taking right now. Happy to answer questions about it.
Yeah, fantastic. And let's sort of like go through and do each of the industry participants, and then we'll, set everyone free to speak together. How about Antoon? Would you like to sort of walk us through what's happening at Imcyse?
Yeah, thank you, Monica. So at Imcyse, we are working with a platform technology based on immotopes. Immotopes are peptides, synthetic peptides that contain a natural epitope, CD4 T cell epitope, that is present in autoantigens, such as in type 1 diabetes, is an insulin-derived peptide. And this peptide has in the area outside of the MHC class II binding groove a thioredox motif. And we have discovered that if you have that motif, then you actually drive a CD4 T cell response into a phenotype of a cytolytic CD4 T cell. We know the cytolytic CD8 T cells quite well, also in viral infections and in cancer. cytolytic CD4 T cells are somewhat less known, but have clearly been reported in literature. And so we have a method to induce these type of CD4 T cells in a peptide and antigen, autoantigen specific fashion. What these T cells then will do in an autoimmune patient, you give these immotopes and you induce these cytolytic CD4 T cells. They will recognize in the target organ, in the pancreas, antigen presenting cells presenting the wild type peptide coming from the autoantigen insulin. And they will, because they have cytolytic capacity, they will kill the antigen presenting cell. And what is also very interesting, they will also kill T cells that have interaction with this antigen presenting cell at the same time. So that can be T cells with somewhat other specificity, but also not only CD4, but also CD8 T cells when they encounter with the antigen presenting cell. And so this is why we call this more immune deletion. We actually take out the bad cells to begin with the antigen presenting cells that stops the downstream activation of these immune cells, but also kill the pathogenic CD4 T cells. And hopefully also quite a number of the pathogenic CD8 T cells. And we've shown in mouse models that this works quite nicely in type 1 diabetes, NOD mouse models, but there's always a challenge to translate those findings to the clinic. We've finished a phase 1 study in type 1 diabetes patients, early diagnosed. And we have some encouraging data there in terms of stopping the progression of the disease to a certain level and for quite a substantial amount of time, as well as the immune readouts, biomarkers of the induction of cytolytic CD4 T cells, and also some reduction in in pathogenic CD8 T cells. Of course, this was a small study with few patients, thus we should really not overinterpret these data. And currently we have a phase 2 study ongoing. It's an impact study in type 1 diabetes patients together with the Inodia consortium. And also happy to answer questions later on if needed.
That's great. That's really informative. Okay, how about let's hear from Francisco at Provention Bio. How are things going, and how are I guess if you could sort of like lay out your approach.
Absolutely. Thank you for having us, Monica. So Provention's approach is a bit different from our colleagues in the sense that rather than targeting antigen-specific pathways, we are trying to target autoreactive cells, autoreactive T cells in general, regardless of what antigen they target. So first, we are trying to prevent the primary breakdown in tolerance that happens when viruses infect beta cells in early stages of life, and then chronic infection leads to tolerance breakdown in the right genetic background that then leads to type 1 diabetes. For that, we have our Coxsackie virus vaccine that is expected to prevent acute infection and then down the road, the development of T1D autoimmunity in a subset of patients. As many as 60% of patients with T1D have an infection by Coxsackie virus preceding the onset of T1D, as you know. And when mothers have immunity against Coxsackie, the offspring has 50% less T1D. So that is our early interception approach or prevention. But for subjects who have a different viral trigger or a different trigger altogether and develop autoimmunity, we have teplizumab, the anti-CD3 monoclonal antibody that turns autoreactive cells into exhausted T cells. Those cells stop destroying the beta cells and have some regulatory properties. Teplizumab, as you mentioned earlier, is now in late stage development. It's been reviewed by the FDA for the delay of clinical T1D in stage 2 patients. And we are now in the final stages of that review process, working with the agency on the comparability of our proposed commercial product with the prior teplizumab manufactured by MacroGenics and Lilly, the previous sponsors of these trials. We are also in phase 3 development in newly diagnosed T1D patients. Overall, the data has shown that in trials, a single course of teplizumab can delay the progression to clinical T1D by a median of 3 years in pre-symptomatic patients, and can preserve beta cells in pre-symptomatic and in newly diagnosed patients. So we are excited about this approach. And I know that later we'll talk about combinations. I think, Monica, you selected a great group of companies because you have the 3 pillars here of the potential cure of T1D. The antigen-specific approaches, the immune approaches that target the autoreactive cells, and then the beta cell regeneration or transplantation that will help us, with our combined efforts, cover the entire spectrum of T1D. So it's a really exciting time, I think, in T1D research.
I agree. And I just note that Matthias von Herrath has just joined us, and so I just wanted to welcome him and say thanks for joining. I didn't know if he wanted to dive in and say a little word about
Morning.
Good morning, and what's happening in Novonordisk. Is there something there that is sort of happening that is working towards a prevention or cure? Do you want to weigh in?
Yes, of course. I'm happy to do so. Can you hear me?
Yes, we can hear you perfectly.
Excellent. We have several approaches that might be of interest. As probably some of you know, we had a large phase 2b trial in recent onset diabetes combining anti-interleukin 21 and our GLP-1s that was recently completed and published in Lancet Endocrinology. And the results were good. We preserved C-peptide. We reduced insulin dose while on treatment. If you ask where these things will be going, right now, and we are all kind of in the same boat there, there's ongoing discussions with the regulators as to which endpoints in recent onset trials should be acceptable to move forward. And notably, the discussions have surrounded whether C-peptide and insulin dose are alone sufficient or whether there should be glycemic endpoints. Personally, I can add that glycemic endpoints in recent onset trials will be very complicated to reach. You will need many patients, in some calculations over thousands, especially with the arrival of CGMs and closed loops. But we have great hopes that down the road, it will be possible to proceed with these promising interventions. Also, Francisco just mentioned them from their perspective, with the C-peptide preservation and maybe insulin dose. So that's one thing we are doing. The other thing we are doing is we are in phase 1 together with TrialNet with an antigenic tolerance DNA immunization platform that expresses proinsulin together with response modifiers in the same plasmid. It's low-dose IL-2, IL-10, and TGF-beta. We hope that we can move this forward after the phase 1, alone or in combination with, for example, an induction therapy with an immune modulator. Of course, things come to mind as close to all of our hearts is the recently successful trial that Provention Bio has run with the anti-CD3. And maybe also to consider is combinations with GLP-1s. So these are the type of things we are doing. I should also mention Novonordisk has a large stem cell program where hopefully at some point we'll see a convergence between making beta cells and islets from stem cells and the progress in the immune modulatory therapies. Because the way I view it is that we will need still some immune modulation, hopefully mild, even if we have immune evasive stem cells, and that these two fields will hopefully converge in this respect. And that's what's ongoing. I hope I didn't go too long. I'm very happy to be here.
Thanks for that update. No, there seems like there's a lot cooking over there at Novo. I guess I would ask, I'd like to ask the people who spoke to sort of think about and talk about how these technologies that you're each promoting and each involved in, how might they work together? And then we'll sort of shift gears and talk about sort of the stem cell implant platform with Drs. Millman and McAuliffe and Atkinson. So what do you, would anyone like to venture, how the technologies we've talked about so far might, show some symbiosis? Is it possible?
Yeah, if I may, I think I agree fully with Matthias that these are complementary technologies where if you are able to give long-term suppression of your immune cells attacking the pancreas and the beta cells with either treatments with nanoparticles or with immotopes or anti-CD3, and then of course do some kind of regeneration. Can be different approaches, but of course beta cell regeneration at some form, either by cell therapy or other ways. That could really be the long perspective forward. But of course, each of these still have to prove themselves individually. I think Provention Bio with anti-CD3 is quite advanced now already. So we might first see some results from those type of combinations coming from Provention Bio, maybe together with other companies doing the stem cell work. So really exciting times for type 1 diabetes. And I think this could be also a good example for other autoimmune diseases going forward. Because if you are successful, we've seen that I think also in the tumor immunology, where tumor immunology is quite old, and different things have been tried. Also in autoimmune diseases, many different things have been tried. But if we start to be successful with one or two approaches, then I think this field could really kick off, and there will be much more resources available for us to do these kind of things.
Can I just ask for a minute? How easy is it to, imagine this kind of cross-pollination between companies?
I mean, I can say
sounds good, but can it really happen?
I think first of all, right now we are all sitting in the same boat, as alluded to, that this whole area of tolerance induction in autoimmunity and in type 1 diabetes and for stem cells needs to be opened up. And once this is opened up, I think in this field in particular, and the analogy to oncology is very good, we will need combinations. And not all combinations will work, and some of it will be trial and error, some of it can be tested preclinically. But I think, like it has happened in oncology, there should be tremendous space for synergies between different entities. And certainly the thinking from our side is that that's how this is going to evolve, and that large companies as well as small companies all going to play fundamental roles in this field to ultimately to the benefit of the patients. And you can envision that you have an induction therapy to reset things, and then a maintenance therapy to maintain the benefit that you have achieved. And I think it's an exciting time, because it feels we are sort of at the cusp that we can actually break into the field, as well as in immune modulation and prevention and recent onset, as well as at the same time for stem cell transplantation. So I find this particularly exciting times, more than ever over the last 3 decades.
That's great to hear. This is sort of a bullish pronouncement. I like that. I mean, of course, I like that. But and it's good though to hear from those that are immersed in industry feeling that way too. I think we could just maybe shift our gears just for a minute, and then we'll come back to sort of talking again together to the technology, that HIPSC stem cells from the Millman lab offer, as well as the Captixbio offer. And I mean, I wondered if Dr. Millman and McAuliffe, would you be so kind as to illustrate your solutions and your work? Do you want to go first, Jeff?
Yeah, sure thing. So we're really focused on not prevention, but instead patients who have very little to no functional beta cell mass. So kind of the other end of the spectrum versus what this conversation has been focused on so far, which is, of course, cell replacement therapy, with the idea being that if we can grow up a large number of secreting beta cells in the laboratory, in this case using stem cells as the platform technology for that, can those be transplanted into patients to essentially replace the dead or dysfunctional beta cells that reside inside of the patient. The important thing, of course, here, in contrast to the earlier discussion, is that this wouldn't be an actual cure for type 1 diabetes in the sense of dealing with the loss of self-tolerance that gave rise to it, but instead is just providing the actual blood glucose control that we desire for patients. And one of the reasons why I'm so bullish on this idea is that we already have a proof of concept of cell replacement therapy working with the transplant, which is the transplantation, of course, of islets that come from deceased donors. Of course, we just don't have a lot of this material available every single year, as opposed to the stem cells. We can theoretically grow up a virtually unlimited number of cells to serve as this source of replacement cells and tissues. So the main questions that are in the field right now is, while there's been many protocols published by my group and other groups for describing procedures for generating and producing beta cells from stem cells, where these cells tend to be pretty immature when you compare them to cells from the body. And so a lot of the focus in the academic literature right now is trying to figure out how to do that in such a way that you can actually have fully mature adult-like cells that come from these stem cells. Another major aspect as well that the field is focused on is really trying to understand the role of the non-secreting cells that are found in the islets in terms of them being beneficial, neutral, or perhaps potentially negative in terms of cell replacement therapy. And that I'm thinking of cells like alpha cells and delta cells, which are naturally in the islets, but we're not really certain that they're very important to have for a stem cell product or not at this point. And of course, probably the biggest challenge in the field right now, which is kind of similar to the loss of tolerance here, but is how do we prevent autoimmune attack and allogeneic rejection of transplanted beta cells that we make from the lab. And there's a whole lot of details that you get into on that one, that if you like. And the two main approaches there, which I think we'll be talking about some of this later on, is encapsulation of them to prevent rejection of the cells, or otherwise genetically modifying the cells to essentially cloak them from the immune system. But there's a lot of development needs to occur with that, obviously.
Yeah, the invisibility cloak is a sort of a holy grail, I think. To try to get them to be able to do these implants. Obviously, Sema, Vertex are now moving forward in that space, but the fact remains that even if you get the implant, you still, as a patient, would still require the immune suppression. So that sort of turns me over to Dr. McAuliffe, who his company is proposing that they have a solution that doesn't require. There's a beta cell implant that doesn't require immune suppression. So maybe you can comment on it a little bit.
Thanks, Monica. Thanks to all the other panelists. There's been some great ideas there so far. Firstly, I'd say it's hard to not feel a little bit like a fish out of water. My PhD is based around knee replacement surgery. I'm an orthopedic surgeon with a particular interest in knee surgery. my daughter was diagnosed with type 1 diabetes a number of years ago, and since then I've been involved in type 1 research. Our group is looking at an encapsulation process, and we're working with the CSIRO polymer unit in Australia. One of their claims to fame is the development of oxygen permeable contact lenses. So they've got a lot of expertise in biological membranes. So we're looking for that cloaking system that can protect the cells from immune attack, but still allow them to both survive and function. So we've got a separate program looking at the polymers itself, also with the University of New South Wales, with the unit there. The orthopedic aspect of it that we're looking at the moment is the use of the intramedullary canal of the long bones. We've known for many years that you can undertake emergency resuscitation for patients via the long bones if you place a needle in there, and that drugs and bioactive molecules that are within the canals of the long bones have almost immediate access to the vascular system. So it closes down the response time, and it's akin to placing a device within the vascular tree itself. There's very large volumes within the canals of the long bones, and there's a long history of placing all sorts of implants in there. So we're looking at a specifically modified orthopedic device that will carry the cells in an encapsulated membrane into an environment that we believe will have a lot of advantages.
Okay, that's very interesting. A different approach. I think sort of there's, I don't know if Mark Atkinson wants to weigh in here and sort of give an overview of what we've heard and maybe invite next question.
So I like this because we've had real diverse thoughts in terms of the group, in terms of your approaches, and again the notion of combination therapies came up. Monica's going to have a couple group questions in a moment to get feedback on, but I wanted to ask one the group, and anybody can answer it. When we talk about prevention and cure, I think to this group, they understand those are largely two different things, meaning what you would do in a recent onset versus an established type 1 versus somebody with pre-type 1 are different. But do people think that we've hurt ourselves a bit in the research community by essentially viewing this as a one-off, or like we're going to develop this and it'll be useful for all applications? Hopefully that question makes sense, and whoever wants to tackle it. Or do we need to be more personalized or precision, I should say, in our approach?
Yeah, perhaps Mark, I can take a crack at that interesting question. I think the answer is going to be we need all of the above. I think we're going to need more generic approaches that target the core of the tolerance breakdown. And we're going to need more antigen-specific and more personalized approaches as well. It's not going to be one size fits all, but while we continue to learn about patient characteristics, it would be detrimental if we were to narrow our scope too much in this early stage of T1D R&D. Thanks to organizations like the one you created, Mark, MPOD, and a lot of the pre-competitive collaboration between all the companies, all the academic centers, we have now a relatively good understanding of the commonality to the process, the HLA, the antigens, the autoantibodies, the T cells. And that gives us a chance to attack some of the nodes that will give us perhaps 50, 60, 70% efficacy, then add additional layers of personalization later to get into deep remission. We recently had a good example of that. Provention works with Precigen, Actobio. They have an antigen-specific approach with a lactococcus that produces pro-insulin with interleukin-10. And they got a nice response as monotherapy, but then combined with teplizumab, the response was much larger, in the order of 70% response in preservation of beta cells in newly diagnosed patients. So I think that's a very specific first evidence that the combination of our approaches is going to take us further into the dream of a cure. And we're very excited about the combination with regenerative medicine approaches. We believe teplizumab can prevent the allo and the auto response of those transplanted, regenerated beta cells. And again, it's going to lead to deeper responses.
Yeah, let me echo what Francisco just said. I think it is quite, of course, in the long-term vision, Mark, I think there will be more individualized approaches. But first, we have to crack the door open to this whole enterprise and get a foot in the door. And once then we sort of see how different entities, antigen-specific, general immune modulation combinations fare in the clinic, I think we will then be in a position, like it has happened in oncology, to tailor these therapy more to individual patient features. I think that's how this is going to go in my mind.
Yeah, it does seem that it's going to be a layered approach for sure. I agree with that. I wanted to see if we could just launch a quick poll to the entire audience as well as the panelists. And this poll is, I'm going to launch it. Would you be willing to get your genome sequenced and or undergo annual screening for T1D autoantibodies to detect pre-symptomatic T1D? A pretty heavy yes, right out of the blocks. Well, maybe some maybes. Does anyone want to comment on why this may be important? Francisco, I think you had, this is something you thought might be interesting to ask.
Well, yeah, I think it's very important to understand the unmet need. We need to understand if family members with relatives with T1D, are they interested in learning if they have the genetic predisposition, if they have the autoantibodies that indicate they have pre-symptomatic disease? Do they want to know? And as we offer options, we soon hopefully will have options to intercept that disease. Is this what patients want or not? Right. So that's why we were interested in that question, because there could be some potential downsides of knowing that you have a predisposition for a disease, some anxiety if there's nothing you can do about it. But hopefully we are at the tipping point when we'll be able to do something about it. And it's interesting to hear that the majority of our audience today think that we're moving in the right direction.
I might just add there that 3 of my children are currently involved in a monitoring program in Australia where they undergo annual testing for just that purpose, not genetic sequencing, but they're on a monitoring program for autoantibodies as well as glucose tolerance testing. So there's a program for that in Australia currently.
I think this discussion is kind of dancing around maybe one of the core issues, which is that we don't have definitive predictive biomarkers for if somebody's going to develop type 1 diabetes or not. And genetics, does increase the likelihood of it, so do autoantibodies as well. I think one of the things that the field could really benefit from, particularly with the work that these companies are doing here, is if we had something much more predictive and determinant if somebody's going to form type 1 diabetes, and then comboing that with preventative therapies would be much more directed and probably better for patients.
That's a point well taken. So I'm going to stop sharing our poll right now. And I also am going to take a question here from Alberto Hayek. A comment on a Salk paper last year showing protection from rejection from xenografts of beta cells derived from human iPSCs with cells overexpressing PDL1 protein. Does anyone, is anyone interested in answering that one? Feels like Jeff would answer that one best.
I'm not quite certain what the question is, but I can comment on it, which is in general what many groups, including Ron Evans, who was the PI for that paper, and other research groups and companies as well are doing, is a combination of deleting HLA typically to avoid HLA mismatches as a mode of immune rejection, then overexpression of some other gene to prevent other means of immune rejection, such as NK cell mediated death. And so in that paper, they chose PDL1 overexpression, which is very commonly done on cancer cells as a mechanism for cancer cells to avoid the immune system. But there's lots of groups have different approaches they're using for that. But I don't know if that's the correct combination or not, or if anybody actually knows what the correct combination of knockouts and overexpressions are to actually create an immune evasive beta cell. But that's the general gist of it, is can we, using now that we have CRISPR-Cas9 that is so efficient in genetically engineering stem cells and other cell types as well, can that be used in combination with a beta cell differentiation procedure to create these hypoimmune or immune evasive, whatever term, there's a lot of different words that kind of describe the same thing, cells, so that you wouldn't need to combo it with a encapsulation device that prevents vascularization into the graft, and therefore provide immune protection to the beta cells and prevent their rejection.
May I ask a question back, because that's an area that interests me very much. What do people think is the evidence that we need to do anything beyond lowering, let's say, MHC on transplanted stem cells? And it's a loaded question in the sense, because I agree that it might not suffice. But the reason it might not suffice is that any cell you transplant, if it dies, and there will be some cell death, these antigens from the cell will then leak and be indirectly presented. And whether this cell is completely immune evasive itself or not, that indirect presentation that might affect long-term graft acceptance might still occur and hamper the long-term graft survival, right? So that's my thinking about that. So I'm not so sure whether, if you lower MHC, certainly it's going to be good. You can reduce immune suppression, and you still have some prevention of NK recognition. But that might be at the end enough. Also, because if you make these completely immune evasive cells, maybe you need to add, from regulatory side, a fair amount of suicide genes to that. But I just wanted to ask this question back, because I find this discussion is quite interesting.
Yeah, I don't know, and your points are well taken, and I think there's, I don't know if I'll say consensus in the field, but definitely a lot of agreement and discussion on the concept of the last point you just made about the suicide gene. If we're going to be giving, making these unnatural cells that have special immune characteristics, having a fail switch, especially for early generation products, is going to be, I don't know if it's going to be a necessary safety procedure, but I definitely would feel a lot better about that. In terms of the other issues that you bring up, again, I'm not an immunologist, and we talk about PhDs mismatching what we're doing. My PhD is in chemical engineering, and so I fell into stem cell biology through a whole other set of circumstances there. But I know enough stem cell biology and immunology to be a little bit dangerous. I don't know the field really understands exactly what is going to be required in terms of exactly what we need to protect the cells when it comes to the immune system. It seems a lot of the focus, again, we don't really do much of this research in my own lab, but talking to other people who do work in the space, they seem very focused on just like the cell recognition part of it. And I haven't really heard significant discussions about the point that you just brought up, Matthias, of cell death. And again, like with islet transplantations, the rule of thumb is that we lose at least 50% of the cells with a beta cell transplantation. I've heard as high as 90% in some estimations as well. And I think that's a underappreciated question that hasn't been seriously addressed in the field based on what I've seen quite yet. So I think it's a really good point you bring up. Thank you.
Yeah, I would agree. You know, that whole idea of the the kill switch in case things go wrong is an interesting one. I wondered if Jessica might weigh in. How might AnTolRx's you know, tools, I guess, intersect with beta, you know, or islet, beta cell or islet implantation? Is there a possibility there?
Yeah, definitely. I mean, so that's actually something that we've thought about a lot, because the antigen-specific approach is one certainly that's helpful in the idea of, you know, stopping the improper targeting of these cells, but it doesn't actually do anything about the existing problem, which is that you have beta cells that have been attacked already and that have been destroyed. And so we've thought about certainly partnering with someone who's doing these beta cell regeneration, because if you just look at either approach on their own, there's significant limitations. If you're just taking the antigen-specific tolerance approach, you have these beta cells that have already been destroyed and that are no longer functional. And, you know, our approach honestly doesn't do anything about those cells. But at the same time, you have these beta cell replacement therapies that aren't doing anything about the ongoing problem of you having these immune cells that are attacking the beta cells and destroying them. So if you think about partnering the two approaches, I think that that could actually be really significantly helpful. And that's why these panels like this are so helpful, because collaborations between these companies really should be happening in order to think about the ways to really best treat the patients. Because I think it's not just these one single approaches that are going to do the best work, but instead dual approaches. And certainly AnTolRx would be interested in collaborating with people as we think about moving forward, for sure. Because there are holes, I think, in everyone's technology approach. And that's why if we work together, we might be able to like fill some of those gaps, I think.
Yeah.
So Jessica, thanks for bringing that up. Again, collaboration between companies is one thing, but I don't know, Monica, if you have the time, interest now. Monica developed a question the other day that I think is fitting and maybe worth some more discussion. You want to pull that up, Monica?
Here it is. It's perfect. Perfect timing. Thanks for highlighting that. So here's our second poll, you know, because, you know, this is, my impression is that the symbiosis that exists between academia and industry and within industry is big but underdeveloped. So I think that, you know, what are the main barriers we have to ask? What are the problems? Let's ask the big question, the tough question. What are the main barriers to industry collaboration that might accelerate a clinical cure for T1D? And I mean, when we think about industry collaboration, we have to appreciate that it's industry to industry as well as industry to the bench and academics. So I think, you know, it looks like there's a real sort of a tie between IP barriers and legal constraints, or focusing on one product or asset versus priority on hypothesis testing and research. And yeah, these are some hurdles that need to be addressed, and I'd be very interested in hearing the panelists' ideas for solutions. Yeah. 37 to 37, IP and the asset.
I can give you a perspective on this. We are, many of us here today and others, we are constantly exploring these collaborations. And I'll be a little provocative. I think the main barrier to actual collaboration is just that we are in early days of T1D immune therapy. There's nothing approved, there's no precedent. It's very difficult to combine two experimental approaches. It's much easier to combine an approved approach with an experimental approach from many points of view: financial, regulatory, ethics committees. If you think about the immuno-oncology field comparison, very pertinent. Once the PD-1 inhibitors got accepted and approved, the field exploded. Today, there are 2,200 clinical trials in combination with PD-1 inhibitors and PD-L1 inhibitors, 2,200, ongoing. We are nowhere near that, right? It's orders of magnitude.
Yeah.
I think it's just a matter of time that this field will reach a tipping point where we can prove to all the stakeholders that T1D is a disease that can be prevented and can be cured by our combined efforts. As Mark said, these approaches are all complementary, and we just need to keep crossing the valley of death on our own and trying to collaborate as much as possible until we have enough critical mass to prove that we can make a real dent on public health, on families and patients who we can help. The will is there, and this field is such collaborative attitude that this field has. This is very impressive.
I have some additional insights, being one of the academics on the call here that has had a decent amount of interactions with industry. And I think just to echo the last point that Francisco said, I think the individual people, like we all do this research for type 1 diabetes because we care, a lot of us have a personal connection to it. And so I think when it comes to personnel in industry and academia, there's a lot of goodwill and a lot of willingness to work together. I think there's a bit of a mismatch in terms of the incentives people who are operating in industry and the incentives of people who are working in academia have, that ends up, at least in my experience, has been a major barrier towards working together. And I think a lot of that was implied in the poll that we just took there. And again, I've had a lot of good collaborations with industry, so I'm not referring to those, and I'll give you a little bit, just to give you a little bit of insights of how it's been for me over the years. That a lot of times companies are used to working with like CMOs or CROs for various things. So when they approach academics, they view them as essentially another like CMO and CRO. It's sometimes also, or if they know about academia, they hear things like, oh, academics volunteer their time to be on a study section at NIH or to review papers and stuff like that. So I've had basically kind of a weird mixture of two types of people from industry coming to me, either expecting me to do kind of service in terms of like working with the company, so basically doing it for free, or again, this is not everybody here, I'm just giving you the negative experiences that I've had as an academic, or if they want to like work with the lab, it's purely from like a CRO perspective with no like working on a hypothesis driven sort of project and something that I can actually get a grad student or a postdoc who needs to be doing hypothesis driven research to be able to do there. Again, this is not everybody. That's been my negative experiences there. And I think it just illustrates that the motivations of an academic and the motivations of industry can be, in my experience, the biggest barrier there. It's not like one is right and one is wrong or something like that. And so if we had some sort of I don't know if it's a matter of like a mechanism to overcome that, or an understanding that allows that both sides need to have to overcome that. In terms of my positive collaborations with industry, it has been a matter of me understanding what they need and the industrial partner understanding what we need and able to come to a mutual agreement there. But that seems to be more rare than the other interactions that I've had with industry.
It's almost like the call for an entrepreneur in residence, like a role, like a real hybrid role, which would be reflected in just what you're asking for, Jeff, just a space that could be created and really expanded for the bridge between the two sort of worlds. Does someone else want to comment?
I think I can give some different perspectives also. From our side, at least, two collaborations that are not only a CRO type of situations are actually quite desirable. So that the industry scientists and academic scientists truly put their minds collaboratively together rather than just having a unidirectional flow of money and then a return of services. I think that's where the field is going, which is good. I think the hindrances are actually cultural aspects that we both sides have to work on and are often not too prominently discussed. They have to do also with expectations. One is the expectation of IP in an undeveloped market like type 1 diabetes, and also revenue. This is still very shaky. So one has to somehow meet in the middle because nobody knows how big the market will be, the different estimates. The other one is in terms of the robustness of projects and what is needed to generate data that then enable clinical trial progression. There we sometimes worlds apart, and I live both of these sites. A lot of it has to do with repetition and money and reagent purity and the drivers, whether you want to do something for patients or you need to do something because you're required to write a paper. So these are things I think we have to work on to bridge this gap. But certainly, I don't think they're insurmountable if everybody is willing to put effort into this.
Yeah, that's well said. It seems that there's a real wish or a desire to actually build that space out more, where the two camps intersect. So that's good news. Mark, would you like to weigh in on that? Okay.
Well, we only have 2 minutes left, and before we thank everybody, I get one last thing about what we've talked about. I mean, the hour's flown by. Does anybody want to say about pragmatic ways to intervene in type 1, prevention or cure? Meaning a lot what we've talked about, and again, probably the few minutes left, but if somebody would summarize with their thoughts on cost effectiveness, the ability to screen populations that came up, who's going to pay for it? I think we haven't really addressed that issue. Does somebody want to pick that one up?
I'm happy to say a few words about it, but I think prevention is very different from therapeutic treatment, and I mean really primary prevention, because you will have to identify the right person that will develop type 1 diabetes and not expose a lot of people to a treatment where they may have no benefit because they would never have developed type 1 diabetes. I'm not sure whether we are there yet, but I think type 1 diabetes of an autoimmune disease gets into that direction, and we also feel that, let's say, early intervention with immunotherapies like driving cytolytic CD4 T cells or Treg cells, it's probably more efficient early on than later in the disease. But from a company perspective, it's easier, I think, to develop a therapeutic medicine or a therapeutic vaccine if you drive for a remission or cure, than a preventive treatment. And also the price you can ask for a preventive treatment will probably be very different than for a therapeutic treatment, which companies will take into account when they start to develop these treatment modalities.
That's well said. We're getting to the top of the hour here, and I just wanted to say, just to be considerate of people's really tight time schedules. I feel like this conversation could go on, and I appreciate everyone's input. I think it was really good to sort of flesh out some of the bright spots and landmines, and I appreciate everyone's input. I would like to announce that together with DKNet and the Signaling Pathways Project Index and the T1D knowledge portal down in UC San Diego, as well as at the Broad with the T2 knowledge portal, the Sugar Sciences created a T1D research-based D challenge using in silico tools. We have some cash awards behind people using this in silico tools to actually develop novel hypotheses. And registration is now open. You can find more information on our website. So thank you all for attending, driving discussion. We look forward to our next interactive town hall style State of the Science scheduled for August 26th, same time. And this one will be titled Targeting the Immune System in Type 1 Diabetes, Which Cells and Which Pathways. Thank you again.